Thank you for tuning in. This is my presentation on Chapter 9 of Jacob Stegenga's Medical Nihilism, entitled Hollow Hunt for Harms. As you can probably tell already, these slides will share the same upsetting artistic style I used for my presentation on the Kruger article. I just hope that my presentation is not as upsetting. I know of an old Chinese proverb that roughly translates into all medicine is at least partially poison. Note that this is a translation due to me, which means a whole lot was probably lost in translation. Some say that this saying goes way back to an ancient Chinese medical text known as Huangdi Neijing. On the right hand side of the slide is the cover of a commentated English version of this text published by Columbia University Press. Uh, Now, of course, by medicine, this saying would be referring to traditional Chinese medicine, which is mostly herbal, except maybe with the occasional non-vegetarian ingredients such as dried seahorses. But ironically enough, Stegenga's Chapter 9 demonstrates that to some extent, this saying seems to apply even more accurately to so-called scientific medical interventions out there today. As an overview, we'll see that various conceptual, methodological, and social factors contribute to the systematic underestimation of harm in clinical research. So now, what is harm? The interpretation of an effect of a medical or pharmaceutical intervention as being harmful rather than beneficial is a normative judgment. Normative judgments are often dependent on values and can be rather debatable. So this, well, brings back a lot of memories from our discussion back in week 2 when we talked about chapter 2 of medical nihilism and the Ereshefsky article about how to define health and disease. We can agree that this is not always straightforward. Stegenga, in particular, brings up the example of methylphenidate, branded as Ritalin. This was, still is, used to treat ADHD. Some overworked teachers might think all kids need this, but if you think kids ought to move around more, you would consider this as purely harmful. In most cases, however, what harm means should be unambiguous from context. And it doesn't have to be a serious adverse effect. Like if you take this drug, it might fix your headache, but you never sleep again. Well, that's the kind of harm that gets everybody's attention. But when it's something small, like making you gain some weight, then the ways in which the harm is systematically overlooked would be much more insidious. There seems to be no way to reliably assess the causal nexus there. So Stegenga points out that physicians simply wouldn't seriously conclude that the drug was what contributed to your weight gain in these cases. Well, another way in which the harm profiles of medical interventions are often obscured is when the conspicuously bad terminological choices kind of cover up severity or depth of the harm. This problem is prevalent in a lot of fields. Finance people, for example, cover up a lot of common sense under jargon so that we think only they can do what they do. Well, at least I think that's the case. It's something I hate very much because it seems just so purposeful. It's not just technical language. It's intentional terminological choices. Um, in medicine, Stegenga points out that this is not as simple as summarizing potential vomiting, upset stomach, or headaches as potential side effects of a drug. For example, when the FDA talked about serious harm such as strokes and death caused by PPAR modulators, they referred to these events as clinical safety signals, and some drugs in this class were removed from the market because of, quote, clinical safety. Well, that just kind of makes it sound like some employee on the production line forgot to wear gloves or something. Now, on the other hand, the underestimation of harm in clinical research is also partially due to the way in which harm is operationalized. For example, some measures of harm are terribly insensitive to many potentially serious kinds of harm, and Stegenga gives the example of the Hamilton rating scale for depression again. Last week we discussed Chapter 8, wherein Stegenga argued that this scale is terrible when it comes to measuring the effectiveness of antidepressants. Now it turns out that there are even worse measuring tools for the severity of the harms of antidepressants, but it is the primary measuring instrument used in trials of antidepressants, which obviously speaks to the terrible large-scale systematic bias. You can see that according to the scale shown on the slide, having suicidal ideation adds 3 points to a patient's score, and the score does not go up unless attempts at suicide have been made. But the deep unsettling problem is that some antidepressants have been found to cause patients with suicidal ideas to develop more frequent suicidal thoughts and even signs of self-mutilation. But this scale is insensitive to both. Neither of these adverse outcomes increase the score. Now, I think this speaks to the same problem of any attempt to assign discrete numerical values to the degree of severity of condition. This set of possible adverse outcomes can never be exhausted by a finite number of categories. So it seems to me that while the Hamilton scale here is a true example of the tragedy of such measuring instruments, its potential insensibility to possible adverse outcomes is just kind of knowable a priori. Let's now look at a more technical argument drawing upon the publication bias behind first-in-person studies, which are experiments where interventions being tested are administered in humans for the first time. Now, I want to be consistent with Stegenga's notations, but I also use this E here to denote the member of relation. So molecule X is a member of the class of molecules of type T, and T is a member of the class of all drugs D. So evidence from a phase 1 trial on X is relevant to the harm profiles of X and T and D. So this is relevant to relation is kind of transitive in that sense. Therefore, evidence from phase 1 trials is extremely important for our knowledge of the general harmfulness of drugs overall. But the problem with publication bias is extremely severe. On one hand, there is no registry of what molecules have been tested by such trials, and because of that, empirical studies of the publication bias of phase 1 trials have relied on records of institutional review boards, and it appears that about 95% of phase 1 trials are not published at all. So why is the publication bias so bad? Well, on one hand, it seems to me that a lot of phase 1 trials are straight up horror stories. They're extremely risky for the participants. I will not repeat some of the details Stegenga provided in the chapter. But let's talk about a deeper consequence of the publication bias issue. Um, this is the more technical argument I was referring to. Stegenga puts this in very precise terms. I will try my best to summarize in a very succinct way. So let's use K to denote the hypothesis that molecule X is harmful, and then E would be the available new evidence relevant to the harm profile of X, but not necessarily in favor of K, and then our conditional probability of K given E represents our assessment of the harm profile of X, but this has to be directly proportional to the prior probability that the molecule X is harmful. This is also due to Bayes' theorem, I think. The ratio would be expressed as the probability of E given K divided by the probability of observing E. So prior probability of K depends on past evidence regarding X, other molecules of type T, and more broadly, all drugs D. So we do not have access to most of this past evidence um, because of the publication bias, which implies that the evidence we do not have access to is more likely to confirm K, than the evidence that we do have access to. So our assessment of the probability of K, the prior probability of the hypothesis, and therefore the assessment of the harm profile of X, which we expressed as the conditional probability given E, are both artificially low. In other words, of all the drugs that are tested for clinical use, the proportion that appears harmful is artificially much lower than is truly the case. Now, it may seem as if we did not need all this formal probability stuff, but think about it. Before we followed this argument all the way, we only knew that we didn't know what we don't know about harm in general, and now we know what we would probably know if we did know what we don't know. Isn't that something?
Now, in the previous argument, there was one somewhat ambiguous point. I phrase it as if we're just taking all of molecules of type T as the reference class for assessing the harm profile of the molecule X, I think. But what if the more appropriate reference class is much smaller or bigger? Should we compare X to some other molecules like it, or all of T, or to just all drugs? But in this particular context, you will see that the harm profile assessment will simply move, in the direction of corroborating the previous conclusion as long as you take into account uh, at least more molecules of type T. Because since the publication biases regarding the harm profiles of T and D are both systematically more prone to underestimating harm, with a better reference class, the harm profile of X would be more accurately assessed, and therefore X would probably appear more harmful than it otherwise does. Now, if we look at clinical trials, um, and how they're typically conducted, we see that they may just be bad at detecting harm in general, or at least the kind of trials that would detect harm more effectively just generally don't happen for kind of obvious reasons. So Stiganga uses the concept of statistical power to illustrate this point. And roughly speaking, um, statistical power is meant to characterize the sensitivity of a trial to detect an effect of the intervention under investigation, when there is such an effect, of course. So the statistical power of a trial depends on the effect size of the intervention, the number of subjects in the trial, and the variability of the data. And the designers of clinical trials usually have the most control over the variability of the data. Given the ubiquity of financial incentives to produce results that many that usually overestimate benefit, um, trial designers almost always have this great incentive to maximize the trial's sensitivity to benefits. But this is, of course, only one kind of statistical power. Benefits and harms are both effects of an intervention. If you are artificially improving the sensitivity to benefit, for example, by including a relatively homogeneous group of subjects, then this leads to lessened sensitivity to harm. For example, in drug trials, it is a common practice to exclude elderly subjects, subjects on other drugs, or subjects with other diseases. We see in the case of statins used to lower cholesterol levels that the risk of myopathy, which can even cause kidney damage and death in severe cases, is much higher in women elders and people with other existing conditions, which means they're usually on other drugs. But these are precisely the kind of people excluded from trials. So this gives us reasons to distrust the results from trials and explains the ubiquity of cases where drugs are pulled from the market after people have already been seriously harmed. So most data we have about the harms of medical interventions come from post-market studies conducted only after the interventions are approved and marketed for clinical use. So these studies are usually observational designs, also called phase 4 trials. The important epistemic consequence of these studies, I think, the consequence of them being observational designs is that even if um, regulators do not always have access to evidence divulging the harms of medical interventions from randomized trials, they could still rely on evidence from non-randomized studies and be confident that, on average, they're not overestimating the harm profile of medical interventions. And this will be further substantiated by some of the practical consequences I'll talk about in a few minutes. Well, I think this is also because, for example, it was found that those who consume medications more faithfully according to their doctor's advice happen to be healthier on average than those who consume medications less faithfully. Note that we should not be committing the cum hoc ergo propter hoc fallacy here. We're talking about evidence that those who are healthier anyway experience better outcomes and fewer harms when they're on the medication. So this is a confounding factor here from one of the examples given in the chapter. Observational studies still tend to overestimate the benefits of interventions and underestimate their harms, even though they are the sort of primary source of data suggesting the presence of harm. Empirical evidence of this was found in a meta-study that compared estimates of harm profiles of interventions by both randomized trials and non-randomized trials. Well, I think this is also because harms of drugs are always unintended, and side effects can be bizarre and unpredictable. So the problem of selection bias is generally less of a worry. There's no good reason to think physicians and researchers bias treatment allocation somehow with respect to unwanted effects. So what advantage does randomization really afford in the search for harm? So on the other hand, for a new intervention to obtain approval to be produced and marketed, the bar set by the FDA is really low, requiring only 2 randomized trials that show that a new medical intervention has some beneficial effect, and you can do a million trials behind closed doors though. But we just discussed earlier that sensitivity to harm is just generally really low in these trials. So one of the practical consequences of the fact that most data on harm come from post-market studies is that regular patients inadvertently become guinea pigs in the grand scheme of things, in the hollow hunt for harms. And moreover, as we have seen in Worrell at first and then repeatedly throughout our readings, the dominant view from EBM encouraging a kind of overvaluation of the evidential significance of results from randomized studies provide very unjust reasons for regulators to undervalue the significance of results of non-randomized studies, which, however, provide most data divulging the harm of medical interventions that we have access to. So one empirical evaluation of this kind of underestimation of harm by post-market surveillance suggests that underestimation rate is about 94%. So, by the way, I think the order in which Stegenga presented these arguments is very good. At first, I kind of wanted to talk about secrecy of data right after talking about the consequences of publication bias of first-in-person studies. But of course, and now it seems this is more appropriately something you would address after seeing almost the whole picture about harm presented here. Now, obviously, the status quo is just atrocious, at least in the United States. In Canada, things may have gotten slightly better since about exactly 1 year ago. I read in a CBC News article from March 2019 that medical devices and drugs that used to be carefully guarded by Health Canada were all starting to be posted online, and this includes previously secret data from clinical trials and other submissions provided by companies that Health Canada uses when it considers approving medications and medical devices. I also read in another article that this had put some pressure on the FDA, but right now I'm really not sure what happened there so far. I thought our friends south of the border just typically don't respond very well to pressure. Now, but in general, in both the United States and in Canada, companies that pay for trials claim that they own the data from the trials, and researchers are bound by clauses in their contracts constraining their ability to share data in practically all industry-sponsored research, even when they suspect harm. And phase 3 trials also suffer from serious publication bias, very much akin to what we saw with first-in-person trials. And moreover, there's so many examples that Stegenga gave here, where the first one with reboxetine, data from 74% of patients remained unpublished. With Vioxx, data showing or suggesting cardiovascular harm was withheld, and with olanzapine, branded as I think Zyprexa, um, it caused extreme weight gain and concomitant diabetes, but the data was withheld for years. With paroxetine or Paxil, there was a massive lawsuit that went on for years exposing side effects including withdrawal symptoms that led to suicide or an increase in suicide rates in children and teenagers who used it. And with Tamiflu, most of the data I think is still being held secret. So I think access to evidence from all medical research should be explicitly legitimized as part of our right to health. So why isn't this explicitly in our Charter of Rights and Freedoms? It should be, but of course this would be something we can discuss more during our online seminar. So now we have this quote from famous FDA epidemiologist David Graham. Safety isn't on the radar screen regardless of what FDA officialdom would have you believe. Scientifically, FDA uses statistics in a biased manner that favors industry at the expense of patient safety. So by the time regulators are assessing the benefit-harm profile of an intervention, the harms of the intervention have been systematically overlooked and underestimated. Regular patients have inadvertently become guinea pigs in the hollow hunt for harm, and then their inadvertent sacrifices in the hollow hunt for harm are overlooked again and often dismissed as lower quality evidence. And we continued to not know what we don't know, even though we now know what we would probably know if we didn't know what we don't know, and we know it's all just so disturbingly problematic at the end that we, or at least I, kind of want to go back to not knowing what we didn't know that we know now. I'm sorry that this is a very sad note on which I end this presentation. Thank you very much for listening to the whole thing. I have had fun making this.